So I presented data from our first-in-human phase 1 trial of the oral first-in-class covalent Werner helicase or WRN inhibitor R07 589 831 in patients with microsatellite instable or MSI and or mismatch repair deficient or dMMR advanced solid tumors. So what is WRN? Well, WRN stands for Werner helicase, which is an enzyme that belongs to the RACQ family and it plays a crucial role in the maintenance of genome integrity. WRN actually serves as a DNA caretaker enzyme, playing a critical role in the unwinding of non-canonical secondary DNA structures such as holiday junctions, bubbles, and forked duplexes, which are formed in response to DNA damage and or DNA repair processes. And RO7 is a first-in-class covalent WRN inhibitor, and this is important because WRN inhibition actually represents a synthetic lethal vulnerability and also a therapeutic opportunity in cancers with microsatellite instability or MSI. And by inhibiting WRN, this actually leads to an accumulation of DNA double strand breaks, mitotic cleavage, and eventually to apoptosis and cell death. And so RO7 actually engages covalently with a cysteine 727 that's located in an allosteric pocket of the WRN helicase. And this covalent binding actually locks the helicase in a single conformation, preventing it from transitioning to the subsequent conformational state that's critical for its helicase activity. So in this clinical trial, we provided the first early clinical proof of concept for effectively drugging WRN. The most common adverse events that were drug-related were mainly manageable grade 1 to 2 nausea, vomiting, and diarrhea that was seen in a minority of patients. We showed that RO7 was rapidly absorbed and eliminated with a half-life of 4.4 hours, and PK was predicted to reach steady state after 14 days. Importantly, we observed that WRN resulted in resist tumor shrinkage that was observed across different dose levels and indications, including 5 RECIST partial responses in 3 patients with endometrial cancer, and a patient with colorectal cancer, and a patient with ovarian cancer. And importantly, all of these patients were MSI patients who were PD-1 or PD-L1 inhibitor resistant. We also observed very durable disease stabilization in the majority of MSI checkpoint inhibitor resistant tumors. And these data are supported by the metabolic responses that we observed in a majority of patients. And interestingly, metabolic responses measured by FDG PET were associated with RECIST PRs and durable stable disease. We also observed early ctDNA reductions in the majority of patients with molecular responses, and we also saw that early ctDNA reductions were associated significantly with partial responses and patients with durable stable disease. Currently, dose optimization is ongoing with 3 randomized cohorts to establish the recommended phase 2 dose.